Ah.
Hello.
Can you hear me?
Yes, we can hear you. This must be Let me welcome everyone to this session, the ERC session entitled COVID-19 Frontier Research in the Spotlight. Welcome, everyone. The European Research Council needs little introduction. It is well known for funding scientists and scholars of any nationality in any discipline to undertake groundbreaking research following a bottom-up approach, and excellence is its sole evaluation criterion. This is a celebration of curiosity-driven approach, free of any thematic constraints. It fosters diversity of scientific inquiry and scholarship. Despite this lack of predetermined direction in terms of subject area, the ERC-funded portfolio by now includes a number of projects with potential relevance to the COVID-19 pandemic. The pandemic itself has had an enormous health, economic, and societal global impact. It's had significant impact on science itself and on the general public's interest in science, the process and its outcome. While there is a consensus that SARS-CoV-2, the virus underlying the current pandemic, is here to stay, we all look forward, obviously, to the pandemic itself being over. I am Magdalena Skipper, Editor-in-Chief of Nature. I am joined today by 4 prominent scientists funded by the ERC, each of whom will speak of their own experience of making their research relevant to a more resilient post-pandemic world. I invite you all listening to our conversation to think about questions. At this point, I'm not certain whether the chat functionality is working, but if you find it working, please do feel free to type in your questions into the chat. And just as a reminder, we have 45 minutes for our discussion. So without further ado, I would now like to introduce our discussants, our panelists. So with me here, we have, I hope that Professor Gupta was able to join us. She is a Professor of Theoretical Epidemiology at the University of Oxford. Her main area of interest is the evolution of diversity in pathogens, with particular focus on the infectious disease agents that are responsible for malaria, influenza, and bacterial meningitis. She uses mathematical models to generate new hypotheses regarding the processes that determine the population structure of these pathogens. With us also, we have Professor Ahluwalia. He is group leader in ultrasound, microwaves, and optics at the Arctic University of Norway. His research interests focus on optical nanoscopy, advanced bioimaging, optical trapping, integrated optics, singular optics, and optical sensors. He leads a cross-disciplinary biophotonics and integrated optics research group that focuses on developing optical nanoscopy and its application towards clinically relevant biological applications. With us also, we have Professor Cerullo, who is Professor of Biological Drug Development at the Faculty of Pharmacy and Head of Drug Research Program at the University of Helsinki. He leads Immunoviral Therapy Lab, an interdisciplinary laboratory promoting cutting-edge science in the field of oncoimmunology and immunotherapy, using viruses as platforms to induce or orchestrate the tumor-specific immune response. And last, but definitely not least, is joining us today is Professor Bartels. She is full professor at the Free University and Medical Center in Amsterdam. She is a psychologist and a behavioral geneticist. She leads a research group that aims to understand and identify the genetic and environmental influences on well-being. Welcome to you all. Can I double check that we have Professor Gupta with us? Perhaps not yet. I think there is a difficulty, a technical difficulty there. I'm going to start, therefore, with the first question directed to Professor Ahluwalia. You have developed different variants of super-resolution optical microscopy, so-called nanoscopy, using photonic chip, which can significantly bring down the cost of this approach. How do you see these technology improvements can be applied in the context of COVID-19? How can they help the current pandemic?
Sure. It was the quest of seeing is believing, they say, and COVID virus has a size of just about 150 to 200 nanometer. Unfortunately, these sizes are below what it can be seen using a conventional optical microscope. We have a conventional optical microscope which costs in the order of, say, 20 to 50k euro, but they can only image to a size up to 200 nanometer. Within my ERC project, what I developed was a photonic chip-enabled super-resolution optical microscopy, which can image structures down to a few hundred nanometer, a few tens of nanometer. And this was at the cost of a standard optical microscope. And this happened in 2019 and previously, and then happened SARS-CoV-2 pandemic. And since then, a group of researchers from Norway and from Germany, we came together to see what can we do to help the cause. Desperate time call for desperate measures. We have a microscope which can be user-friendly with a photonic chip, but we still have the cost, which was approximately a factor of 10 times away. And thereby, we started looking at technologies such as mobile phone-based technologies, where we can use cameras for mobile phone, and we can use laser printing to do print the microscope and use our photonic chip to image the super-resolved optical microscope. Just to give you a glimpse, as you can see in the slide, on the left side, a conventional optical microscope can image structures down to 200 nanometer. It can cost only 50k euro. And the Nobel Prize was given in 2014 for a discovery of super-resolved optical microscope, but it came with a cost of $1 million. This, however, was not possible to be widely used. In my ERC application, I proposed that instead of using a very complex microscope to perform laser beam shaping, I use a smart mass-produced photonic chip, which can send the light to the sample directly. These photonic chip works with the very similar principles of optical fiber. And these photonic chips can be retrofitted with any standard optical microscope, converting into a super-resolution optical microscope. And by doing so, the price was reduced down to a few $100,000. However, after SARS-CoV-2 pandemic, we realized that this was not good enough, because these microscopes or advanced microscopes have to be in biosafety cabinet where we have viruses. Thereby, we have come up with a strategy of completely re-engineering the microscope by using a laser printer to make the microscope body, as you see on the right corner, use consumer optics, consumer electronics such as mobile phone, because your mobile phone has all the processing power of your computer, and it has a camera, which can be used to replace a conventional microscope cameras. And finally, we integrated our photonic chip, and on the footprints of 20 centimeter, we developed a super-resolved optical microscope with a price tag of less than $1,000. And we have imaged it we have shown its demonstration for imaging SARS virus, which is a size of few hundred nanometer. And this is a bottom-up approach and a good example of how fundamental research can lead to situations which we not originally planned for.
That's really fascinating. So when you were originally working to make this technology, if you like, more accessible, both from a financial and practical perspective, what was your original idea? What would be the application that you originally envisaged before COVID-19 came on the scene, as it were?
So before the COVID, my initial impression was to produce even more advanced microscope, which will still have the cost effectiveness, but it will be more advanced in terms of throughput. For example, all of us can sit today and do a chat from sitting in our home because of the bandwidth provided by photonics, by optical fiber. And the same kind of impact I wanted to bring of photonics, of photonic chips to the field of microscopy, where the microscope will be more advanced in terms of resolution. They will be catering higher throughput, which means that they can image large areas at smaller time scale, and therefore can be able to apply it for applications where diagnosis is of interest, which was very different from a COVID application.
Yes, exactly. But it is remarkable that you were able to take this work and think about repositioning it. Do you now? So, how do you imagine it's going to be used? I mean, you say you demonstrated that you can image the virus particles with your particular technology. Do you envisage that the application here will be mainly for basic research, but now at multiple locations, which are of course capable of containing the virus? Or do you envisage that there may also be a diagnostic use to this technology?
I think it's perhaps both. At least for the pandemic situation, we were quite early to understand that this war is going to be long. There will be a need of developing affordable tools which people can use at point of care. And it's no more to constrain the virus in only one part of the world. It has to be addressed globally. So a technology has to be with a price tag which can be globally affordable. But having said that, we also feel that this will have applications in markets such as pathology or scanners, where the method has to be based on decision making. So there we need much more complexities in the microscope with AI involved, and in that aspect, I believe it will have also application in the clinical side.
Thank you. Very interesting, and again, so interesting to see the diversity of applications in a way that can make a real difference. So I'd like to turn to Professor Sunetra Gupta, who has now joined us now, and I believe you can hear us now. There's sort of slight technical.
I'm so sorry.
We're solved. Absolutely fine. Wonderful. Great to have you with us. So I would like to turn to you and ask you to tell us. So your main research focus on pathogen evolution is, of course, very relevant to the current pandemic, broadly speaking. And your theoretical work on pathogen epidemiology can have direct implications for vaccine development also, something we all eagerly await in the context of the current crisis. So I wonder if you can share some details of this work with us, how this theoretical work can directly influence our direction with vaccine development.
Absolutely. Thank you so much for that opportunity. Do I get the slide, or is this, are we doing it without the visual aid?
There you go.
We have the slide. Perfect. Okay, so in a nutshell, what I've been working on over the last 25, 30 years is a kind of theory, a kind of unified theory, if you like. It's a bit grand. Okay, but just to try and understand how pathogens evolve, particularly those that exist now or co-circulate as a set of multiple strains. So that theory yields many testable hypotheses, and one of them was with respect to influenza. We developed a new theory of how influenza evolves, which we call the antigenic drift model. And in the process of testing whether this theory was correct, which we did with a combination of serological surveys, that is to say, to look for antibodies in the blood of people who'd been exposed to this virus, and structural studies, we managed to home in on a new way of making an influenza vaccine that would cover against the entire diversity of strains of influenza. Again, very, very briefly, what we did, the theory predicted that on the surface of influenza, there would be some regions which were not entirely invariant, but very limited in their variability. We call these the epitopes of limited variability. So it's like saying influenza may have lots of hats, but it only has 4 shirts. And so that was the theory, but then Craig Thompson, who's shown here, he took this theory and identified those epitopes of limited variability in influenza, and we have now licensed that technology to Blue Water Vaccines, a company in the US, and we're developing it to make a vaccine, a single vaccine, which should protect us against all strains of flu in all subtypes. So now that set us up. So this pipeline that we've developed, or this overarching kind of activity that our lab engages in, going from theory to the development to translation, has actually put us in a very good place to tackle or try and understand this epidemic. So José Lorenzo, who has been involved in the theory, and we've developed a set of models which try and provide the context for what's going to happen in the epidemic itself. So how can we use the limited amount of data that we have to understand how deadly it is, where it's going, how many people are likely to be immune, what we can infer from rates of seropositivity in the population, what is the effect of people already having resistance in the population. So all of these are questions that José has been answering, and we have a few preprints that address these very critical issues. Craig, meanwhile, has been using his expertise that he developed while working on flu to develop assays that probe for antibodies against SARS-CoV-2. And this is just an example shown here of using that novel methodology of making a pseudotype virus that is decorating an HIV virus with the spikes from the SARS-CoV-2 virus, and interrogating the blood of people to see if they have antibodies that neutralize that pseudoparticle. We've been using that technique now both to look for prevalence of how many people have already been exposed to SARS-CoV-2, but also to see whether the other circulating coronaviruses provide some protection against this new virus. So that's in a nutshell how we've harnessed our expertise to try and address the important issue of where we're at with this really problematic virus.
Thank you. Really fascinating. I love the analogy of a virus wearing many hats but fewer shirts, and that's what you are focusing on in order to find this or pursue this vaccine approach. But of course, can you say a little bit about how different SARS-CoV-2 is from an influenza virus? Of course, it's a new type of coronavirus. How much do we understand about the dynamics of its evolution? Is it comparable? How does this analogy of changing items of clothing work for a coronavirus as opposed to an influenza virus?
Well, so that's an excellent way of approaching this problem. So that each particular coronavirus, each type, so for example, COVID, the one that causes COVID-19, would appear to be much less like, in terms of its wardrobe, have a much more limited wardrobe. So each coronavirus kind of sticks to its own wardrobe. It doesn't have the luxury, like flu, of saying, okay, tomorrow I'm going to wear the red shirt. It just can't do that. So that's quite useful in the sense that making a vaccine, this is why I feel have confidence that we will be able to produce a vaccine towards this virus, because it's not going to suddenly turn around and change and be antigenically very different. So at that level, it's much more like measles than flu. But where it's different to measles is it seems that it's likely, and if you compare it to the other coronaviruses as well, that the immunity that you get when you experience measles, which is lifelong, that's not similar. That is not what happens with coronaviruses. You tend to lose immunity and get reinfected every 3 or 4 years. And the most parsimonious assumption about this virus is that it will behave the same way, that we get immunity, then we slowly lose it. The next time you get infected, you suffer less disease. But in fact, what happens most likely is that your immune response starts to focus more and more on what is specific to that coronavirus. But where the analogy might be useful about sharing or, looking for epitopes that are of limited variability is that it's possible, we think it's more than possible, we think it's very likely that all 4 circulating coronaviruses share some items of clothing with this coronavirus. So if you think each coronavirus has its own wardrobe, but there are common bits that they share within their wardrobes. So ultimately, we should be able to exploit this particular pipeline to make a pan-coronavirus vaccine. So that's one thing that we are quite seriously pursuing, although at the moment, of course, what we really need is something to fight the current threat. So yes, I think that's something a future goal, but I wouldn't want to compete right now with the pressing urge to make a vaccine against the current virus.
Of course, understandably, but it's an interesting and actually a very positive goal to work towards, because of course in recent years we have seen other local epidemics of coronaviruses. So the concept that we may be in the future going towards a pan-coronavirus vaccine is indeed very promising. And in the context of, again, running with your beautiful analogy of the extent of the wardrobe of the pathogen here, it does sound promising in terms of the development of vaccine sooner rather than later. I just have one final question for you, and that is, of course, we're dealing with a pandemic, so very clearly we're talking about a global scale here. The work that you talked about in your laboratory, is it done on global populations? Are you mainly looking at a UK infected population? Can you comment on that? And
well, in fact, very much so. And in fact, what we've been doing is, we've been looking at what samples, the samples that we've had made available to us are from the Scottish blood donors, the transfusion service, which is the data very quickly flashed up. So that's been great. And we've also been working on a cohort, a set of samples from an antenatal clinics in Oxfordshire. But that's just because that's what we've personally been working on in the lab. But at the same time, we've been exporting the technology to a variety of different regions. And Craig Thompson has really been working, flat out, because it's one of our goals to export, to transfer this technology to various settings so that this neutralizing assay can be rolled out in different parts of the world. So we're doing that actively, and we're also trying to set up a consortium, which we've called Co-Zero, which is again a global consortium that looks where we can report various people around the world, various groups can deposit their results, but also where we can have a conversation going about what the different results mean, because sometimes most people use different techniques. So given the variety of techniques and the range of data that are out there, to make sense, to come up with a coherent framework for how this is spreading, what we can do about it. In order to do that, it'd be very useful to have this international platform. And so I'm working with people to try and establish that.
I'm so glad you mentioned the importance of global collaboration and exchange of methods and data on a global scale. And I guess there is no other time like the pandemic that focuses the need for that kind of collaborative work. Thank you. I'd like to turn now to Professor Subunit. So your work has focused on cancer immunology, including tapping into this potential of viruses as vehicles to induce and orchestrate tumor-specific immune response. And for cancer, these approaches present a very promising line of treatment. So tell us a bit about your work, and what about COVID-19? It's a very different disease after all.
Yes, sure, it's a different disease. So basically our work, as you said, has been focused on cancer, and how can we use viruses to orchestrate the immune response, as you said, in order to trigger an anti-tumor immune response. The discovery we have done, which is part of also my ERC grant, is that actually we can use viruses to vehiculate within the tumor-specific antigens in order to divert the immune response that is usually against a virus to be against the tumor. So what we have done, we have taken very normal, common viruses like adenovirus, and we have decorated these adenoviruses with tumor-specific antigens, with the tumor pieces. Now, when this adenovirus gets back to the patient, our immune system, or the immune system of the patient, is very used to recognize an adenovirus, so it starts a very strong immune response. But because the adenovirus is now decorated with tumor pieces, with tumor-specific antigens, instead of having only an anti-adenovirus response, we have rather an anti-tumor immune response. And as you said, this technology...
I know you have
yes.
I know you have a slide. You're illustrating it very beautifully with your hands, but I don't know whether you need a slide.
Well, I do have a slide. Yes, fantastic.
Perfect.
So basically, if you can see, thank you very much. Thanks, Magdalena, to remind us. So if you see on the upper part, that is exactly actually what has been so far our technology for cancer. So basically taking the pieces of tumor, basically, which we call technically tumor antigens, we slightly modify these tumor antigens, and in only 15 minutes, we create this very stable complex that when it goes back to the patient, creates a very strong anti-tumor response. Why did we come up with this idea? I was obsessed with rapidity and affordability. I wanted a system to be fast and cheap. Fast because tumors mutate very fast, and cheap because I really wanted to see this technology to be translated into human settings. And in fact, a phase 1-2 clinical trial will start in a few months for cancer. Now, during this year, I've been always thinking of infectious disease. I mean, can we apply this technology for infectious disease? Now, although viruses can mutate rapidly sometimes, but I always thought, they mutate rapidly, but not as fast as tumor does. So I was always under the impression, which was wrong, obviously, that this technology would not be very useful for pathogenic disease, for pathogens, until last March, when the pandemic really started to pick up, and then I started to think, whoa, if only we had a vaccine that could be developed in minutes, in months, and could be very cheap to just be tested. So I start to think, together with my group and some also key people in the company we funded a couple of years ago for the cancer vaccine. We start thinking, can we adapt this technology to this anti-COVID-19 vaccine? It turns out that, of course, the majority of the vaccines that are now in clinic, they are vaccines that are targeting the spike protein of the coronavirus, which is a fantastic target. It generates very strong antibody response against the spike, eventually also some degree of T-cell response against the spike. But obviously, the virus has a lot of other different targets we might eventually want to target. in a very rapid and fast and cheap way. So we start to develop this peptide technology. We start to translate it into COVID-19 vaccine. We have actually identified some very specific part of the virus we can add in minutes on the surface of already developed vaccine in order basically to go to the clinic as fast as possible and to make the existing vaccines better. So this has been more or less our story of last couple of months, and how this ERC project has been turned from anti-cancer vaccine into anti-COVID-19 vaccine. And hopefully, I know we have fantastic preclinical data, and hopefully we will make the difference. Who knows?
It's so encouraging to see that effectively the system you describe is really a modular system. And you say it's fast because initially you were thinking about the rate of mutation, but here also it enables you to assemble these molecules much more quickly. And so, yes, effectively the approach becomes versatile. You mentioned in the context of the cancer vaccine that phase 1 trials are about to begin. First of all, can you remind us, just in case, what phase 1 trials are designed to test? And can you comment, where do you envisage we are at with this approach for COVID-19? When do you think the clinical trial will begin?
Yes, from the cancer point of view, with the clinical trial, we start in 4 different indications or 4 different cancer type, and basically by using an oncolytic adenovirus decorated with very tumor-specific antigens, and that will start as early as May, and it will be a clinical trial. Basically means that the first time real human beings, real patients will be tested, will be treated with our virus, and we will very carefully look at toxicity and a little bit of efficacy. And that's what basically are going to do in May in triple negative, in breast cancer, lung cancer, melanoma, and sarcoma. That's for the cancer type. If we move this to COVID-19, what I really wanted to do with COVID-19, I just wanted to make available something that, in my opinion, was fast and useful. So our strategy has been just approach a company that are using already, that have developed already an adenovirus-based vaccine and are already in clinic, and actually offer them, our help, identifying, some other specific targets of COVID-19, and to make sure that their vaccine will just be bigger and broader, and hopefully, not and hopefully better if this is necessary. That's been as simple as that. It's just being approaching people that were already in clinical trial, and ask them whether or not they will be interested in our approach.
Thank you. I'm sure we will come back to the question of vaccines and immunity. There is at least one question that has come in from an audience, but I will hold it for afterwards, because now I want to turn to Professor Bartels, who has just disappeared from our session. So I will actually wait for her to rejoin us, and I will ask you the question. So there is a question that came in from somebody in our audience, and the question is really fundamental. Are there vaccines that protect better than an actual infection? The question goes on to say, for example, with a common cold, you can get it over and over again, but there is no vaccine that can help. How will this be like in the case of COVID-19? So the sort of naturally acquired immunity versus vaccine-generated immunity. Can you comment?
Do you want me to comment? Yes, Vince.
Go ahead.
So
maybe if Sunetra wishes to add anything to it.
Okay. So usually, or at least what we teach, in class, usually there is nothing, more. There is nothing better than normal viral infection. It gives you a very good protection because the different arms of your immune system are all engaged. So you have a very good antibody response, a very good, T cell response. So it's a little bit broader. The case of COVID-19 is a little bit peculiar because the immunity of this virus seem not to last for a very long time. Generally, people that are developing vaccines, especially what are technically called the subunit vaccine, what these people are doing are taking a common virus and they are putting within this common virus the spike protein of COVID-19, and then use this common virus genetically engineered to express the spike protein of this in order to be a vaccine. Now, this might be, and might be, we don't know it, and we won't know for quite some times, but this might be one of exceptional case where the vaccine will give you, I don't want to use the term better, but probably a longer protection than the real immunity. This can happen with the viruses that are not as immunogenic as other viruses, either because they are immunosuppressive, they have some kind of tricks to be immunosuppressive, or because they mutate all the time. So this can be one of the cases. I hope this is the case, but we won't know before a couple of months.
So would you like me to say something? Exactly. I mean, that is. Sorry, can you hear me?
Yes, we can.
Yeah. Okay. Great. So it is true. That is the holy grail, isn't it? I mean, I always teach my students the vaccines that we have are the low-hanging fruit on the vaccine tree. They are where we've been able to mimic natural immunity. And they're also the ones that induce very strong natural immunity and long-lasting natural immunity, like measles, mumps, rubella. All of these things are childhood infections. They give you lifelong immunity upon natural exposure. And our job as vaccinologists has been just to try and mimic that. And we've got almost, we get there almost up to that point. With other vaccines, we have a real problem. But that's what people are doing. They're looking for clever ways of inducing immunity that's better than natural immunity. We've done that for something like the pneumococcal vaccine, where we conjugated the glycoprotein and made the capsular polysaccharide and we can induce a response which is way better than the natural exposure. So it can be done, but it is a big ask. So with coronavirus, we're sort of in between the kind of measles paradigm and as I've previously, flu one, where the virus itself changes, or HIV, where the virus changes within the host, or other viruses, herpes viruses that hide in the host. So we're somewhere in between. We do make an immune response, but it probably isn't long lasting. So the real question, though, is do we want people to die? We want to stop people from dying. So we want to try and make a vaccine that gives enough immunity that stops someone dying at a particular point in time. Maybe they'll go on to be reinfected, but hopefully that will simply, boost their immune responses and keep them alive. So I think that's the goal. That's the goal.
Very useful to have that situation of coronavirus in the spectrum of different viruses. So often when we talk about COVID-19 and the current pandemic, we focus on the biology both of the virus and of its host, i.e. us. But we also talk about something else, and we talk about our lack of well-being, mainly during the pandemic, during the lockdown. So I want to turn to Professor Bartels, because your research is actually to look at the other end of the spectrum, if you like, right? So looking at the well-being and that interplay of the genome and the environment that underpins that. So as I say, we tend to focus on those who have struggled during the lockdown and in the face of the global crisis more generally. But what about you have seen that it's important to look at those who not only cope, but actually perhaps thrive? Tell us a bit about that work and how it's applied in the current context.
Yeah, thank you. Well, in my research, we collect a lot of data to understand the causes of differences in well-being. And we were just running this project and collected a lot of data till February, when suddenly the pandemic hit us all around the world. And we quickly stopped that particular big survey that we did send out to over 200,000 individuals, and created a new one where we ask a lot about COVID-19 and the effects for people, but also well-being. And what we observe in these data, because we have now longitudinal data in normal times and during the pandemic, and we see that about 15% to 20% of the people actually increased in their well-being, and they were more optimistic and had more purpose in life. And we're now trying to understand why. And the main aim, of course, is to understand why that happens, to help those in the end who suffer. It's not the aim to make everybody happy, but it's to help those who suffer with the data from the people who do well. And indeed, normally we tend to forget the positive outcomes of things like this, because the effects are devastating for many people. But we also tend to forget to learn from the people who do better under these circumstances.
So indeed, fascinating to think about it. And you said you're trying to learn what it is about this cohort of people who thrive under these conditions. What kinds of parameters are you looking at?
Well, we do everything in a genetically informative design. So we look at the genetic predispositions for well-being, for optimism. But we also look at if COVID-19 was in the family or not. We look at socioeconomic status. And we do everything with database linkage. So we link our data from the Netherlands Twin Register, for example, based on postal code, to many environmental variables. So we know a lot about the socioeconomic status of the environment and also about green spaces in the environment. We link it to the data that we have been collecting over the past 25 years. So we know a lot about these individuals. And hopefully we can learn what made them more resilient. Some people fluctuate a lot. Some people are very stable. And the question is, if stable people stay stable, or is it the fluctuating people that are better to cope with these circumstances that are very unique?
And remind us, please, how global is your data set?
The data set currently is European, but we are collaborating all around the world because these kinds of measures are collected in many places. Not every design is as optimal as ours, because we were in a fortunate situation that we were collecting just before the pandemic came and were quickly adapted. But many longitudinal genetically informative data sets exist all around the world. And of course, we want to compare all these differences, because every country is different and every individual is different.
Absolutely. And it's so interesting to see that the pandemic puts a particular filter and focus on your research, because you say you started the research before the pandemic itself. But I sort of anticipate that the current events will bring it into sharper focus and perhaps give it, I don't know, stronger results, maybe? Yeah.
Yeah, it's actually a unique design that has very devastating effects, of course, for many people, but also has unique opportunities for research.
Exactly. All we have time for, I want to thank all my panelists very much. We've heard fantastic stories examples of how fundamental research can be applied at this time of the pandemic. And thank you to all of you for listening to us today. Thank you. Bye.
Yeah, thank you for having us.
Thank you very much. Bye-bye.
Have a good day.
Goodbye.